Hello and welcome to the Transgenic Games, where we change the genetic information in poplar tree DNA to produce different traits. This has been going on for over a decade now. What we're actually doing through processes such as one called CRISPR-Cas9 is we're removing genetic information from part of the DNA and inputting different information so that the trees will do one of several different things. Let's look at 4 different things that these trees can actually do. These genetically modified trees, number 1, can have accelerated growth. They're a commercial product. Over 39 million acres of poplar are actually farmed in the world today. They're valuable. They're also valuable for potential biofuel use. That can be a good thing. For the economy and for the environment, we have potential upside. This accelerated growth, however, can also contribute to certain dangers, which we'll get into later. Now, second, greater carbon capturing abilities. When we have more massive trees, when they grow faster, we're definitely going to be capturing more carbon. So that makes sense. That's one of the goals of these transgenic poplars. If carbon in the atmosphere is a potential problem, if, say, global warming is a potential problem and is aggravated by carbon in the atmosphere, this can be a good thing. Number 3, pest resistance. There are certain beetles, for instance, which kill a lot of trees, including poplar trees. We're experimenting on poplars. This has also been done in other breeds, where pest resistance can be extremely improved with genetic modification through these processes. And number 4, producing no isoprene. Isoprene is specifically produced by poplar trees in their leaves as a stress response to, for instance, dry summers. Now, what this isoprene does is it reacts with emissions from fossil fuels, that is, cars driving around, for instance, which creates ozone. That reaction creates ozone. It goes into the atmosphere. Again, we come back to potential global warming issues. If global warming is an issue aggravated by ozone, then this reduction of isoprene, this removal of it in genetically modified trees, can be a great upside. Those are some of the goals of transgenic poplar trees.
Okay, to get these desired traits, we mix genes from completely different organisms together to produce the traits that we want. So for the poplar trees, we might be splicing in DNA from bacteria to acquire pest resistance in the poplar tree. A highly successful method of gene editing is the CRISPR-Cas9 method, which we'll talk about in a little bit. And then the Golden Gate cloning is an example of how we splice in new DNA. So it's used for the assembly of multiple DNA fragments in a defined linear order. This method is specifically found in plants. So genetic editing is essentially biological coding. DNA from other organisms are spliced into the poplar DNA to acquire desired traits, such as the bacteria DNA to acquire pest resistance. All right. CRISPR stands for clustered, regularly interspaced, short palindromic repeats. This method is derived from the immune system of bacteria. So bacteria have nucleic acids, which find the inserted viral nucleic acids and attach to them. So in the CRISPR-Cas9 method, the guide RNA behave the same way as the nucleic acids in the bacteria would to find these specific genetic patterns or codes. And then Cas9 is a protein that removes the targeted DNA sequence. So right here you can see the guide RNA is finding the desired genetic material, and then the Cas9 protein is simply removing it. And then you can splice in whatever desired genetic material you wish. So here's the different components of CRISPR-Cas9. We have the guide RNA right here, which is the targeting mechanism, which finds the genetic material, and then the Cas9 protein, which functions as molecular scissors. So we can program the guide RNA to find specific sequences. So in the poplar trees, we could remove specific DNA and insert the bacterial DNA that would make it pest resistant. Cas9 can break the covalent bonds in the sugar phosphate backbone of the DNA, and then the donor DNA sequence would be inserted in replacement.
I'm going to talk to you today about the goals of multi-site genome editing of the Populus tremula and the Populus alba poplar trees. As we talked about before, we use the CRISPR-Cas9 and the Golden Gate kit technique to target multiple gene sites. This is unique in the poplar tree. Many other genetic modifications focus on just editing one gene site at a time. In this experiment, we are targeting multiple gene sites simultaneously. To do this, we are using 2 single guide RNAs simultaneously to target multiple gene sites at the same time. We're experimenting on the efficiency of these methods and corresponding results. For the target genes, we target both single gene mutants and multiple gene mutants at the same time. For single gene mutants, we modify the short root gene to see if we can produce the hairy root gene instead. We target the short root gene at the PTA-SHR site, and the result was that 21 out of 22 alleles were genetically modified, which is a very high result. We also target 2 mutants by using 2 single guide RNAs simultaneously. We did this in 2 specific instances. We targeted the YUC4 and the PLT1 genes simultaneously. The result was that 3 out of 4 tested produced biallelic mutations. We also targeted the LBD12 and LBD4 genes. The result was that half of these, or 2 out of the 4, produced biallelic mutations, which is a very high result. This image shows the PTA-SHR gene sequence site. They used CRISPR to target this gene sequence and took it out and inserted the desired gene sequence instead. And these are 3 examples of the mutations that occurred out of the 22 alleles. This image shows an example of resultant double mutations by targeting simultaneous sequences of YUC4 and PLT1, as well as the LBD12 and LBD4 genes. These give 2 examples of the 2 resultant mutations that occurred. The CRISPR-Cas9 with the Golden Gate mode cloning method was an extremely successful method. As well as the results were that at the PTA-SHR site, there was deletion across both target sites, or small deletions or insertions of nucleotides. At the YUC4 PLT1 site, as well as the LBD12 and LBD4 sites, we saw that small deletions or single nucleotide insertions were at single gene sites.
Hello, I'm Bella, and I'll be talking about the ethical implications today about polar trees. So obviously we each recognize that there are clear benefits to this genetic engineering, and the first being that if we believe that the climate and that the globe is becoming warmer, these trees would work as kind of carbon absorbers and would work to kind of cool the earth down a little bit. So in doing this, carbons absorb better into bigger and older trees, so we would use, as we've just talked about, the CRISPR-Cas9 and all of the technology from that to implement trees to become bigger and older at twice the speed. So obviously this would work as a solution to reduce the carbon in the atmosphere, and that would be something great as a benefit. However, we also have downsides, which are there are negative effects. The first being that while CRISPR-Cas9 is highly effective, it is not perfect, nor is anything really in science. So with this still comes mutations. We don't know exactly what those would look like for each tree, but we do know that they would most likely be harmful. So anytime that we are using genetic engineering, we would have to be very mindful of that. Secondly, genetic engineering has an effect on biodiversity. When you insert a bunch of trees into any kind of an ecological system, we don't know the effects that it will have on other organisms. We do not know if it can be invasive to other trees. In addition to this, the more trees there are, the more chance there are to have larger fires and possibly admit to this problem, which is already here. So it's not a perfect solution, and there obviously are negative effects and ethical problems that come alongside it. Additionally, the big question is, when do we stop? We know that genetic engineering is possible today, but now where are we going to find the limitations to this? Politicians like Justin Trudeau are using this as part of their political campaigns because it is such a heavy issue, and it is so powerful in changing the way that we go about our life, and we can change the natural world. So we have to decide at this point when we are going to use this and who is going to limit when we use this. While it comes with great benefits, it still has clear negative effects on the environment that are stated previously.